Moving a liquid from point A to point B typically requires either a sloping surface or a pump of some sort to apply pressure. A new kind of material that is in early development requires neither, instead relying on a squirming skeleton of microscopic fibers to move it in a direction, opening the way for a class of fluid capable of worming itself through a channel. Hello friend. My name is Yukash Kimura and today I am going to tell you about kind of liquid that can push itself along a flat surface. If you not subscribed our channel yet, then please subscribe. Let's start. Researchers from Brandeis University in Massachusetts took a hint from nature and investigated how the biomechanical properties of materials called microtubules could be applied to a mixture to make it move in a single direction around a container. Anybody who has watched a microscopic amoeba stretch out its loopy arm to pull itself under the cover slip has seen microtubules in action. Most complex cells, and even a few bacterial ones, contain a network of microtubule fibers collectively called a cytoskeleton, which is responsible for giving the cell its shape and transporting materials around as they twist, bend, shrink, and stretch. These fibers are made up of a string of proteins called tubulin, which spirals around on itself to form a cylinder about 25 nanometers across. In this particular case, the researchers used the microtubules found inside the nerves of a cow's brain. On their own, the mass of fibers would be about as impressive as wet cobwebs. But the researchers found they could turn a watery mixture of cow microtubules into molecular motors by adding a couple of other ingredients. The first was kinesin, a protein which naturally attaches to the microtubule and walks along its length in a molecular waddle. Another component was the energy-carrying molecule adenosine triphosphate, ATP, which generally provides a kick of power whenever it donates one of its three phosphates to proteins such as kinesin. Putting them together, the researchers found the kinesin connected a pair of microtubule strands like a rung on a ladder, and when powered by the ATP walked them in opposite directions. Each kinesin that walked off the end of a fiber was soon replaced by others connecting other fibers, creating a squirming mess of microscopic worms. Check out in the clip below how these writhing strings of protein slide against each other with help from the kinesin. The interesting thing about this swirl of fibers was that it produced small whirlpools in the gel-like mixture. This turbulence could be harnessed to push the fluid in the same direction simply by choosing the right shape for the container. The researchers stuck to containers they could map mathematically, such as discs and donut-shaped toroids like the one in the picture up top, finding by picking the dimensions of the container the messy churning of the fibers became a steady current in a single direction. While this only works in containers with precisely the right dimensions, it does scale up, meaning liquids can be encouraged to flow over a number of meters. Of course, once the ATP runs out of phosphate to hand over to the kinesin, the flow grinds to a halt, which would make it unlikely for large-scale transport of liquids. However, there could easily be a role for self-propelled gels in the future of mechanical engineering. From a technology perspective, self-pumping active fluids set the stage for the engineering of soft self-organized machines that directly transform chemical energy into mechanical work, the researchers suggest in a summary of their paper. Even without an application, this research provides insight into the dynamics of moving fluids inside our own cells. Perhaps one day we'll be seeing wet robotics powering our cybernetic body parts with self-propelled fluids fed by our body's own reserves of ATP. This research was published in Science. Thanks for watching. Please subscribe for more interesting videos.